My name is Sky Lee. I'm the assistant director here at the Genomics Core at the Anschutz Medical Campus, and it's an honor to give you kind of a background and some information about what we do here at the Genomics Core. So, without further ado, please join me in exploring the wonderful world of the Genomics Core. So, our mission here at the Genomics Core is to help you navigate through the treacherous unknowns in your scientific exploration that involves a next-generation sequencing readout. So you need to address your hypothesis, but you have a limited budget. You don't have specialized personnel, and you don't know which technology to use. Or in other words, you have an idea, but you're unsure about the best way to execute your idea. And of course, there are no guarantees in life, but what we can do is help maximize the odds of your experiments succeeding. And to achieve that, basically, here's how it goes. Like the core itself was established in 1999, so over 20 years, right? And we are the largest genomics core facility in Colorado, and we have trained personnel with decades of experience to handle your samples and deliver data. We invite field application scientists and manufacturers' technical support to advise your experimental designs. We negotiate reagent discounts with the manufacturers on your behalf, and we have a collection of the latest state-of-the-art technologies available to you, and our flexible workflow is compatible with your needs. And, just to kind of give you an idea of what this looks like when the samples come to us. And like I said, this is really under the hood stuff. I'm sure most people don't know what's going on, but this kind of gives you an idea of preview, right? And so let's say we start here, the sample drop-off, right? Now, this can vary what kind of sample it might be, just because we have a wide range of services that we offer. And I'm going to go into more of that in the later slides, but it could be as simple as just DNA or RNA that you've isolated from your tissue or cells, something of that sort, and you just want to look at the DNA or RNA profile from that sample, right? But we also have single-cell sequencing. We have spatial transcriptomics, which is the latest kind of cutting-edge technology that we've adopted here. So these are the samples that we would kind of intake, and you could say that's step number one. And of course, you have to coordinate with our staff to make this happen. And, your samples will undergo preliminary QC, and if it all passes, we go through a lengthy process, which is, in this flow chart, step 1 to 2, that's kind of the longest point, but, just for the sake of simplicity, this is what it looks like. And after all of that is done, the one thing that everything here has in common is step number 2, which is the next-gen library prep. So, all these different sources, whether it's DNA, RNA sequencing, or single-cell spatial, the common criteria is that we're going to make these DNA ultimately compatible to be sequenced on the Illumina platform, which we call libraries, okay? And these libraries will also undergo quality control because these instruments, they're very expensive and they have a very high, strict criteria for what is acceptable to be sequenced. And so assuming that it all passes our QC, it gets sequenced. And that's what it is. Step 3, QC. Step 4, the next-gen sequencing. And after that's all done, we generate FASTQ files for you, and we send that off to you as well as your bioinformatician. And I know I'm kind of going over everything at a really fast pace right now, and some people might be wondering, like what exactly is next-gen sequencing? So that's something I'd like to kind of go over as well. I'll give you a brief introduction as far as what next-gen sequencing is about, and hopefully everybody will be up to date. And the one thing I'll kind of point out too is that a lot of the stuff that I'm going over here in this talk is actually on our website as well. So if you wanted to kind of look at all these material on your own time, at your own leisure, at your own pace, all you have to do is, the simplest way is to Google Anschutz Genomics Core, and we should be the first option on the search list there. So what is next-gen sequencing? Well, let's say that we're starting with DNA fragments, and these could be coming from any source, right? It could be genomic DNA that we fragmented, or perhaps it's cDNA that's been reverse transcribed. But like I said, they're all different sizes, and this could be the starting material. The first thing that we do is essentially we size select the DNA so that they're all the same size, and this is done with magnetic beads. Afterwards, and this is the really important step here, we ligate read primer targets, barcodes, and Illumina adapters. And, just to kind of interject here, if I had to kind of summarize what next-gen sequencing is, it's a process. And I'll just kind of iterate that point. Like what I'm going through this process can kind of be summarized as what next-gen sequencing is. So like I said, we got the DNA that should be in fairly uniform size. We ligate these read primer targets, barcodes, and Illumina adapters. And what we'll do is that we'll flow these at this point, we would call these libraries, and we would flow these libraries across the flow cell. And these flow cells, they have these these little oligos that are covalently bound to the surface of the flow cell, and it's going to capture the adapter that's complementary to these oligos, kind of like Velcro, right? And it'll undergo that first polymerase reaction. So if you can see my mouse, that's what's going on here. And it's going to copy this fragment. And what you're going to see is that you have now the copy over here that's actually covalently bound to the flow cell. Afterwards, this gets washed away and it undergoes another polymerase reaction, but this time, this fragment, this library portion that's covalently bound to the flow cell, it folds over onto itself, right? And then another portion of the, another oligo that's also on the flow cell, it will essentially have a polymerase reaction going in this direction. And this is called bridge amplification. I'm sure you can understand why it's called that. So after this process is done, we have two copies, right? Or essentially they're the same fragment, but they're copies of each other, but just in reverse complement, that are covalently bound to the flow cell. And it'll undergo that process several times. And ultimately what you're going to get is something called a cluster. So sometimes, you know, you might hear the words reads interchangeably with the word cluster, and that's why. So you're going to have these clusters generated, and all the sequences in this cluster are going to be the same sequence, basically. And at this point, what happens is that you have, the adapter portion is what kind of allowed this portion to happen, but then the read primer portion is where these primers will bind, okay? And remember, all these libraries, they have the same Illumina adapters, and they also have the same Illumina read primer targets. So you have this read primer target bind. And what's interesting here is that, as you can imagine, you know, there's 4 nucleotides, right? A, C, G, T. These nucleotides are going to be fluorescently labeled. So you can think about, let's say it's at the red fluorophore, yellow fluorophore, green fluorophore, blue fluorophore, depending on what it is. So first things that's going to happen, you're going to have, let's say the G over here, it's going to have all these fluorescent nucleotides that gets, you know, flowed across, and the C is going to bind to the G, right? And at this point, it's going to fluoresce red, and it's going to take a snapshot. So now we have a picture of this flow cell, and you can have a red color essentially associated with that spot, and that gets either quenched and or washed away, and it goes through the same process again. This time, the next sequence is A, so the yellow fluorescent T nucleotide is going to bind there, and it's going to take another snapshot, like another picture, and then it's going to be quenched or washed away, same thing, and then another T, let's say another yellow, this time green, red, blue, and ultimately here's what this might look like. So this is happening all at once on that cluster, and remember, they're all the same sequence, so this is going to amplify the fluorescent signal. And here's what a representative image might look like. So in the clusters here, it's going to have blue on one image, and then maybe it'll be a different color, and so on and so forth. You can kind of think about it like a flip book cartoon, right? As you flip it, you can kind of see the motion in the cartoon, and that's what's happening. We're going to have many, many, many of these pictures essentially taking snapshots. And when we start looking at each snapshot at a specific point, that's where we can kind of infer like what the sequences were. So, you know, like here, there was a blue fluorophore, I think that was A, so it'll be A, maybe the next one, it'll be a different color, maybe it'll be yellow, and then we know that the following sequence will be a T, and so on and so forth. So that's how next-gen sequencing happens. And there's many different flow cells that are compatible with the Illumina platform. The most common one that we use is called the S4. This is just the biggest one that we have, and this is kind of what that would look like if you zoomed into the flow cell. And after we actually do the sequencing, we'll generate FASTQ files. This is essentially what we'll deliver to you and your bioinformatician. And of course, like the bioinformatician will use that information to essentially, you know, compare it to a reference or just de novo assembly and get that overall sequence from. So that's kind of the background about what next-gen sequencing is. And with that said, we have a lot of different sequencing options available for you, ranging from whole genome sequencing, whole exome sequencing, ChIP-seq, which for the most part is, you know, interchangeable with cut and run, cut and tag, so we can do all of that for you, and RNA sequencing. And again, all this information is on our website, and for the sake of simplicity, I'll just kind of go through one of these, just RNA sequencing, just kind of a representative workflow of what this looks like, and you know, kind of the useful information that's on our website. So let's say RNA sequencing. So RNA sequencing reveals the quantity and sequences of RNA in a sample at a given time. So RNA-seq indicates which genes are actively expressed and to what extent. So let's follow the green arrow. Let's say you're interested in the RNA-seq. Well, even before you begin, the first thing you're going to have to do is consult your bioinformatician. Ultimately, it's going to be a triangle, like essentially 3 people involved, like you, me, and a bioinformatician. And we're all going to be talking and working together to make sure the experimental design is appropriate and whatnot. But even after that, you have chosen your sequencing experiment. You should seek consultation for your bioinformatician to get specific sequencing parameters. Okay, so following that green arrow, this kind of gives you an idea of how to prepare your samples. And really, for RNA-seq, you should have a minimum of 3 biological replicates and a control. That's not required, but it is recommended. And, our most popular service is the mRNA prep, which is the first one here, which is the poly A-selected mRNA prep. So this will give you the transcriptome profile of your sample. And if you want to do that, what you're going to have to do is prepare 500 nanograms of total RNA and 50 microliters per sample. So over here, this tells you exactly what you need to prepare in order to get your RNA sequencing done. And afterwards, once we have the sample, and assuming that your samples pass our QC, we will generate libraries from your sample. And in my previous kind of workflow, like the what is next-gen sequencing, we kind of went over how those libraries are made. So we do that, and based on the sequencing depth that we have chosen, you have chosen, we will sequence it. And you might be wondering, okay, well, what is the sequencing depth? Well, first of all, that's another thing that your bioinformatician can help you with. But even here, this kind of gives you some idea of what some representative read depths are. So let's say, once again, following the green arrow all the way down here, for DNA RNA sequencing, the read metric refers to the sequence from a single library fragment, and we perform pair-end reads, which means that we're sequencing from both ends of a fragment to generate high-quality alignable data. And so that's what the reads mean. And for mRNA, the standard is 40 million reads per sample. So if you're not sure where to begin, this is a great place to start. And again, it really depends on your specific experiment. Like if you wanted to look at alternative splicing, for example, the 40 million wouldn't be enough. You would have to go over 100 million reads per sample. But again, that's the thing that you would talk to your bioinformatician about, but this kind of gives you a quick roadmap as far as what you would need. So, you can tell us we want to do 40 million reads. Afterwards, we would, at this point, load those libraries on the next-gen sequencing platform, the Illumina sequencer, and it would be sequenced based on the depth that you have chosen. And once again, we'll generate FASTQ files. We'll deliver that to you, to your bioinformatician, to do downstream analysis. So that's a really simple example. So that pertains to DNA and RNA sequencing. But like I said, our core, we're always at the forefront of introducing new technologies. And this one's kind of in the middle ground, so like the bulk RNA, bulk DNA sequencing, that's kind of the first generation. Let's say there's a second generation, which is single cell sequencing. Well, what is single cell sequencing? Typically, that's interchangeable with single cell RNA-seq. There are single cell DNA sequencing available too, but in the vast majority of cases, what we're looking at is single cell RNA-seq. And as the name implies, what we're talking about is getting the transcriptome profile at a single cell resolution, right? So in the previous example, we're talking about bulk RNA-seq, where you may have tissue or cells that you've essentially disrupted. You got the total RNA extracted from all of that, and we'll sequence that to get the transcriptome profile from your whole, your mixture of samples. But here you're going to get the transcriptome profile at a single cell resolution. And to achieve that, this is how it works. So basically, the thing that's special here are these gel beads. And you can think about these gel beads kind of like, you have all these oligos sticking out of them. And these oligos, the two features that are really special about them is that they have these barcodes, and you can think about them as cell-specific barcodes, and I'll just call this a green barcode for the sake of simplicity. And you have the poly-DT portion, and you can kind of imagine the poly-DT portion is the portion that hybridizes to the poly-A tail of the mRNA. And again, these beads have all these oligos sticking out. They're like kind of like hair sticking out of your head. And all of these oligos are going to have the exact same barcode. So you have a green bead that's going to have a green barcode, and you have a yellow bead that, let's say, to have a yellow barcode, a blue bead with a blue barcode. And of course, the one thing that they all have in common is that they have the poly-DT portion. And what's essentially, let's say you bring us cells, essentially cells suspended in PBS. and you want to do single cell sequencing. So we would load your cells simultaneously with these gel beads onto the instrument. And the goal of this, what this instrument does is essentially it's a microfluidics instrument that captures one cell and one bead in this little micro droplet surrounded in partitioning oil. Okay. And so kind of go that in the reverse direction. Ultimately, the final product will be, we'll have a tube of oil, and inside you're going to have these little micro droplets, all these little tiny little droplets. And inside the droplet, the goal is to have one cell and one bead in it. Okay. So this is what we call the single cell capture. And once that's done, the goal is to have those gel beads, which have those oligos, undergo reverse transcription. So the poly-DT portion is going to capture the poly-A tail of the mRNA, and that'll undergo cDNA synthesis. And that cDNA is going to retain that barcode. So remember, it had a green barcode, right? So every single cDNA that's generated from that microdroplet with the green bead is going to have a green barcode. And, to use the specific example here, all the cDNA generated from this cell over here is going to have a red barcode. And all the cDNA generated from this cell over here is going to have a blue barcode. And the cDNA generated from this cell is going to have a purple barcode, et cetera, et cetera. And at that point, the gel beads are dissolved, all the cDNAs are generated and tagged with the exact same barcode. Yeah, like I said. And at this point, we pool them all together and then we sequence them. Of course, there's an intermediate step of generating the libraries, but at this point, bioinformatically, what you can say is, although everything is being sequenced at once, we're going to have that cell-specific barcode associated with it. So, you might get a sequence. You'll say, the bioinformatician might look at it and say, hey, I see a gene here, and this gene has a green barcode associated with it. That must mean it came from cell number 1. And I see this other gene that has a red barcode associated with it. That must mean it came from cell number 2. And then, I see another gene that has a green barcode associated with it. That must mean it came from the first cell along with this gene. So you put that all together, that's where you kind of get that single cell information from. And, that's a super simplistic overview of everything. With that said, when it comes to single cell sequencing, we actually have many, many options available as well. So most commonly, people are interested in the transcriptome, the gene expression profile at the single cell resolution, but we also can do immune profiling. And what I mean by that is we can get B cell receptors, so antibodies, or T cell receptors along with gene expression at a single cell resolution. And obviously this solves a huge problem when it comes to bulk RNA sequencing. I mean, let's just take, BCRs and TCRs. They have a very specific alpha and beta pair, that are going to be specific for the antigen. And if you lose that alpha beta pair information, everything else becomes essentially useless. So preserving that information is very, very important. So if you want to get your alpha beta pair for the T cell receptor or B cell receptor, we can do that for you. We can also do some epigenetic studies at the single cell resolution. So ATAC, single cell ATAC, which means that this will allow us to investigate chromatin accessibility at the single cell resolution. And multiome, which essentially is simply put, is doing the gene expression, the mRNA profile, along with ATAC, the chromatin accessibility, simultaneously from the same cell. So that's really amazing. The technology is amazing, right? And once again, just kind of to keep things simple, our most commonly used service is gene expression. So I'll just kind of follow that in this flow chart. And, if you're ever interested in anything else, I'll just remind you that all of this information is on our website. So by all means. So let's say you just follow this kind of blackish arrow down. And the first thing you would have to do is schedule your drop off. So when it comes to all these kind of cell work, it's not as simple as just dropping off DNA and RNA, because for that, we can just store it in -20 indefinitely. But, when it comes to these single cell studies, is a lot of criteria for your sample that has to be met. And so timing and logistics becomes a very important issue. So one thing that's really important is contacting our single cell capture specialist to schedule your sample drop off 1 to 2 weeks in advance. And let's say that our schedule is open, we are open to taking your samples. Well, the next thing you got to do is actually prepare your samples. And here are some important criteria that you would need from your samples. So minimal clumps or debris, high viability at a specific concentration, in a very specific buffer with no EDTA or magnesium. And that sounds like a lot, and it is. But for the most part, it's pretty simple as long as you just flow sort, fact sort your cells before bringing them over. I mean, that will address the clump and debris issue. That'll address the viability issue because you can sort for viable cells. And you can obviously put it in a very specific buffer, like the PBS with no EDTA magnesium. So, you just FAC sort. The only thing at that point you would have to worry about is concentration. So really, it's not as bad as it sounds. So let's say you can do that and you drop it off, and we would at that point undergo preliminary QC to make sure that your cells are actually good. Essentially, the two things that we would look for is viability and concentration. And if it doesn't look good in either of those cases, we would just say we should probably stop here. Like, we shouldn't proceed forward. Like, it's a very expensive experiment. You're not going to get good data out of like poor quality cells. So we'll just stop here, and we're not going to charge you for anything for that. So this is a very important QC step that we would have before proceeding forward. So assuming that your samples pass QC, you would still have to choose your sequencing depth, just like for next-gen sequencing for DNA RNA-seq. Here it's a little bit more straightforward because these are pretty much standardized. So for gene expression, it's commonly known in academic standards that you want to do 50,000 reads per cell. So that's pretty straightforward. And assuming that we capture 10,000 cells, which is the kind of the upper limit of the number of cells we can capture per sample, 10,000 cells, 50,000 reads per cell, that means we'll do 500 million reads for your whole sample. So that's a very easy calculation that we can do on our end. So at this point, the capture happens. And remember, in the previous flowchart, I already went over what is single-cell sequencing. So the capture happens, those micro droplets, reverse transcription, cell-specific barcode, yada, 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 generate libraries. And then we would sequence it. So your libraries are loaded onto the sequencer. And, we sequence based on the depth, like I said over here. So 10,000 cells, 50,000 reads per cell, that would be 500 million. And then at that point, we would generate FASTQ files. And once again, we'll deliver your FASTQ files to you and your bioinformatician. And I will interject here that for single cell sequencing, bioinformatician, that is really, really important. I doubt, unless you're experienced, that you can really analyze your data on your own. So having a bioinformatician ready, even before starting the single cell experiment, would be really, really important. So moving on to spatial transcriptomics, this is like the newest thing that you can possibly imagine. And this technology is more or less at its infancy, and it's still in development, but we here at the Genomics Core really want to be at the forefront of bringing new technologies available to our investigators. So we're still getting our feet into spatial transcriptomics. Whether or not it's perfected, it'll probably never be perfected. It'll just keep on evolving as time goes on, but it's still really awesome, and we want to be there early on. So and many of you might be wondering, what is spatial transcriptomics? Like, I've never heard about this before. So let me just kind of go over what that is about. So spatial transcriptomics, there's actually many different technologies and instruments available, but I'll just kind of go over one that's simple to understand. And, if I had to kind of put it in one sentence, what spatial transcriptomics is, it's getting the transcriptome profile at a spatial resolution, right? So, single cell sequencing is getting transcriptome profile at a single cell resolution. Here, you're getting the transcriptome profile at a spatial resolution. And putting that metaphor, the analogy, I think it's useful even to kind of comprehend what spatial transcriptomics is all about in the context of this Visium assay. So like I said, let's kind of go over this. The starting material, one of the most important thing here is respect to this slide. And it's a very special slide. It's very expensive. And you see that on the slide, there's little 4 squares, right? And essentially these little 4 squares is where we're going to overlay your tissue onto. So if we kind of zoom into the little square, what you'll notice is that on the square, there's going to be these tiny little spots, okay? And these spots, what they are essentially oligos sticking out of them. And these oligos are going to have here a spatial barcode, okay? Again, these oligos, just kind of like that single cell experiment, you have the spatial barcode, which is kind of, you can say, a spot-specific barcode or maybe a space-specific barcode. And you're going to have the poly-DT portion, okay? And once again, using that same analogy, you can say that this spot has green barcodes, this spot has blue barcodes, and, this spot over here will have red barcodes, et cetera, et cetera. And essentially, like I said, these will undergo cDNA synthesis. So the poly-DT portion will capture the poly-A tail of the mRNA, will generate cDNA, right? And that cDNA is going to have that green barcode here associated with it, right? And the cDNA generated from this one is going to have a blue barcode associated with it. So so essentially at the final stage of all these things, you're going to get spatial information from. But I know I'm getting ahead of myself, so let's kind of follow along here. So we also have the sectioning instruments. Essentially, if you bring us a tissue block, we will put it on the instrument, we'll cut the section here, as you can see, and we will transfer this tissue section on top of the little square right here. Okay, and at which point here you're going to get, I didn't say this earlier, for spatial transcriptomics, you're going to get two sets of data out of it. The first one is an H&E stained image. Okay, and this assay is compatible with immunofluorescent stained images as well. This is not something that we would do at the core, but I know I mentioned earlier in the slide that we have a very flexible workflow. So you can do the immunofluorescent staining on your end, and then just kind of work out the logistics to drop off your slide to us, and we will do everything downstream. So that's how you can do immunofluorescent staining and the spatial transcriptomics at the same time. But by default, we're going to do H&E stain and imaging for you when you bring your tissue samples over. So the first, we have that H&E stained image, followed by the actual transcriptomics experiment, which here, the stained and image, slide is over here. We would actually permeabilize it with an enzyme. And, one thing that's kind of not mentioned here is that with that permeabilization is dependent on the tissue. So we do have an optimization step that happens before. So we'll find kind of the time sweet spot, the optimal time that we would permeabilize it for to allow the tissue to be not too degraded, but permeabilized enough so that the mRNA can actually permeate through the tissue onto the slide, right? So let's say that's all good. The tissues are permeabilized, the mRNA will kind of flow through and get to the slide. And here, like I said, we do cDNA synthesis, and that's kind of what you see here. These oligos are going to act as the primer for cDNA synthesis and make a copy of that mRNA. And as you can see, kind of like what I mentioned before, you have this cDNA that has a green barcode associated with it, and you have these two cDNAs that have blue barcodes associated with it, and so on and so forth. And at which point, these are going to be washed off the slide, and then we're going to generate libraries, and these libraries, once again, are going to retain that spatial barcode. We're going to sequence it. And, hopefully I don't want to kind of reiterate this point, but any gene that has a red barcode associated with it, you can say, oh, that came from this spot over here. Or maybe we'll say, okay, well, I'll just use this example. We'll say, oh, it came from all the way over there. And I see a green barcode associated with it. That means it came from that spot all the way over there. And so, there's 5,000 different spots for this Visium slide. That means that you can have up to 5,000 different spots that you can assign the spatial information from. And so here's what the data would look like. Here's the H&E stained image. Here's a kind of a visualization of the transcriptome based on UMIs in this case. So this is kind of like more of a QC image. But here's kind of something that's really interesting, the Visium spot clusters. So I'm sure many of you have seen kind of like the PCA plots, the principal component analysis plots, where you have UMAP or T-SNEs, and you see all those clusters. Well, you can kind of think about this as the same thing, but now we can actually assign those clusters onto the tissue. So that's really cool. Like you're getting the spatial information the spatial information for the transcriptome. And the one thing I'll kind of note here is that everything I've went over here is a representative workflow for the simplest spatial experiment that we have, which is the 10x Visium platform using fresh frozen sample. But this assay is compatible with FFPE samples. We also have another instrument called the Genomics, NanoString Genomics, and that does something totally different. And I see that, I'm going to kind of rush through this actually, just to keep things simple, because I think I'm going a little bit over my time here. So once again, how do you choose your spatial omics experiment? Essentially, with these two spatial technologies, you can think about Visium as a discovery approach and NanoString Genomics as a targeted approach. So let's just choose Visium for the moment. You're going to get, like I said, two orthogonal data types, classic histopathology high resolution image, which means that H&E stained image, as well as a gene expression profile across the tissue. So following the blue arrow, you would have to consult your bioinformatician, and you would have to bring us fresh frozen tissue embedded in OCT or FFPE tissue, right? And the one thing I kind of should point out is that those little 4 squares on the slide, they're 6.5 by 6.5 millimeters. So you should just be cognizant that your tissue size would have to be within that little square. And then afterwards, we'll do the spatial transcriptomics experiment. That's basically what I went over in that whole workflow chart. We'll have the libraries prepared. We'll sequence them on the Illumina sequencer. And then afterwards, you're going to essentially get a FASTQ file from us that your bioinformatician will analyze, and the image file. So yeah, I mean, that's spatial transcriptomics in a nutshell. And the last thing I'll kind of point out is that, like the instruments that we have available at the Genomics Core is the other half of our what makes us so special. So I kind of went over like our spirit of the Genomics Core, where we really want to help you with your experiment, move things forward, and just kind of all the background that goes into how that works, as well as the decades of experience that are associated with all of these. And then we have, like I said, the state-of-the-art instruments that I'm kind of showcasing here. And this doesn't even include like the new stuff that we bought this year. And so actually like this nice looking image here with all these instruments, maybe it should be double the amount here, but I digress. But the other really important thing that I want to point out is that we have free consultation services available here. So these consultation services, they last from 30 minutes to an hour. And during these meetings, we can help choose the experiment that is specifically appropriate for you. We can help you customize your experiments. We can have specialists and tech support from manufacturers to either help you design your experiment or troubleshoot if something went wrong, and we can see what we can do to work that out. We can provide a price quote for you based on your specific parameters, and connect you to a bioinformatician if you actually don't have one. And this is so obvious, but I don't know why some people don't do this. Like I said, this is free. Like you can meet, go over your experiments, go over what you would need to do to get your samples prepared. I mean, the saddest thing that I see is when people have these really, really precious samples. They prepare their samples badly, which, they didn't have to. They could have just talked to us first and they say, hey, I have this precious samples before I even touch them. What do you think I should do? So at this point, it's like, okay, well, let's invite some people, some of these manufacturer tech support over, their field application scientists over. Let's get their input. Let's get your bioinformatician's input. Let's have all these different people looking at this problem from different angles and come up with the best solution for you for free. And I don't even know, like, come on. Okay. So I really highly encourage everyone to just be aware of this. Please let everybody know we do free consultations. So that's I'll just say the icing on the cake. So with that said, thank you very much for your time. Like I said, all the information that I went over is on our website. So if you want to go over it in more detail, please visit us at the Genomics Core. Just Google Anschutz Genomics Core. And if you have any further questions, feel free to email us at the genomics.core@cuanschutz.edu. So thank you very much.
And I do believe that some people have some questions for you, Sky. Some people posted questions in the chat. Michael were two of them.
Yeah.
One of them was, what is the size of the square for the spatial analysis?
Yeah, I mean, so I went over that. So for the Visium, that's going to be 6.5 by 6.5 millimeters. Yeah, and then again, they're coming up with new slides that are going to be 11 by 11 millimeters, right? And for the genomics technology, which I didn't really go over much, if you're interested, just go to our website, look at that a little bit more, and feel free to email me. I'll go over that. For that one, the slide is the section, the area that you can put your tissue on is 14 millimeters by 35 millimeters, so much bigger. You can actually put multiple sections on that slide. So hopefully that addresses that question.
Madison, I can ask my question myself. So just keep in mind, Sky, I'm just talking a little bit medical imaging question. So as you said, it's also transcriptomic. You have your field of view of 12 millimeter to 30 millimeter, but you have only one slice, right? So very thin one. If you do multiple cross slices through the tissue and you acquire those images, is it possible for you to do three-dimensional IHC staining? This is what very hot now in mass spectrometry imaging and obviously in medical imaging.
I would say, I mean, look, if you have infinite budget, the short answer is yes. But realistically, not really. I mean, like I would envision like using that Visium example. I mean, if you had $1 million, you can, you can get like a bunch of those slides, let's say 100 of those slides. And then, these sections are 10 microns thick. So you could put like the first 4 sections onto the first slide, and then the next 4 sections onto the next slide, and then so on and so forth. And then, theoretically, I think it's feasible, but really, practically not really. Like each slide, just the reagent portion of the slide is $5,000. Okay.
Well, then I think the technology needs to improve, because the heterogeneity of the tumor, having only one slice doesn't give you any serious information, right? So so hopefully it will be possible in the future. Thank you.
Yeah, no, absolutely. And, the one thing I'll point out is that, like I said, we've been around for 20 years, but next-gen sequencing has not been around for 20 years. We've been around since the microarray days, right? So you can maybe even think about spatial transcriptomics as microarrays, and then maybe 10 years from now, what Natalie was saying, just kind of like spatial transcriptomics at multiple, kind of the Z axis, that could be available maybe 10, 20 years from now, but I can guarantee that we'll be at the forefront in that respect as well.
Yeah, and thanks so much. It was a great presentation. Thank you.
Thank you very much. Just briefly, what is the resolution of spatial transcriptomics, the spatial resolution? For the Visium slide right now, each spot is, I think they're, 50 microns. Yeah, okay, so Gal already said it, 50 microns, and they're also spaced out by like 50 microns. So you can kind of think about these as 10-cell resolution for each of these spots. So they're not quite single-cell resolution yet. There's workarounds where some people do single-cell sequencing to generate those initial PCA plots, clusters, and then do the non-single-cell resolution Visium and assign those single-cell clusters onto the Visium spatial tissue. So that's kind of a workaround, but I can also tell you, before even what Natalie suggested with the Z-axis, the next stage of spatial transcriptomics is going to be single-cell resolution spatial transcriptomics. So that's kind of like what's going to happen next. I'll tell you right now. All right, so thank you very much. Please feel free to reach out to us if you have any questions.